Welcome to Psydactic Residency Edition. I am Dr. O'Leary, a fourth-year psychiatry resident in the National Capital Region. This podcast is about neuroscience and psychiatry. I do it because, well, I love the topics, and it helps me to learn. I hope whatever you find here helps you to learn as well. I want you to know, though, that, I don't have any editors looking over my shoulder telling me whether I've made mistakes or not. I try to tell you the truth, but the truth is in the eye of the beholder, and I am the beholder for you. Also, please don't mistake what I'm saying here for anyone else's opinions, like the opinions of, say, the Department of Defense or the federal government. Today, I want to talk about something that I have been learning about recently. For years, I've been trying to understand networking and how our brains actually function. And I was given a paper recently from one of my attendings, and the topic of the paper was attractor networks. I found it fascinating, and so I wanted to incorporate some of the insights that I got from this paper into the podcast today. Also, I wanted to let you know that I'm trying something a little bit different. In the past, I've always had a basic script written for the podcast, but this time I only have some notes. So I'm trying to do this without a script. It may sound a little bit different than it has before, but I find if I can do this without a script, then that would actually help me in the future to make these podcasts a little bit more frequently. So if you haven't figured it out yet, I am not a data scientist. I am not a scientist who studies networks. I am a psychiatry resident, but I'm going to do my best to try to explain how attractor networks are an important concept going forward in psychiatry and neuroscience. I want to start, though, by talking about evaluating knowledge in general. Like, how do we know what we know has value? And I think one of the things that is most important to consider is something called plausibility. So plausibility is just, well, this could possibly be true. For example, it's not like breaking any laws of physics or anything that we know from science a priori. And it also helps if the concept can be mathematically modeled, because a model can demonstrate that the proposed concept is plausible a priori. I mean, just because a model works in a computer doesn't mean that the model is actually going to work in real life. But having a model in a computer that says this is how things are expected to function makes it possible, at least, that they could function that way in real life. Another thing that's really important to consider when you're thinking about new kinds of knowledge is whether or not it has something called consilience. So consilience is a concept where different sciences and ways of understanding the world kind of agree with each other. They have concepts that don't obviously contradict a concept in another way of thinking. So when you have a bunch of things together that have consilience, it doesn't prove that it's true, but it makes it a lot more likely that it has at least some kind of underlying plausibility. I think this is especially important in psychiatry about evaluating knowledge. Is there's this kind of I call it halfway reasoning. It's that instead of trying to figure out why things work or testing the mechanisms that we propose for the various things that we do, we just do clinical trials. And this is good because clinical trials can say whether or not we actually want to use this. Is it efficacious? Is it as good as or better than something else that we're doing? But clinical trials do not produce a probability that results are real. The p-value you get at the end is not a probability that the results are real. Instead, clinical trials produce like an estimate of magnitude of effect and whether or not this is likely to be from a random sample of some sort of assumed distribution. So if it seems like it's random, then you don't reject the null hypothesis, which is that everything's just random. But if it looks like it is actually not, that there is an effect of the treatment, then you would reject the null hypothesis. However, this does not say anything about the actual probability that things are real. What we need to do for that is something called Bayesian reasoning. The way to test for confidence that the magnitude of the effect is real, then you have to convert the results of clinical trials into something called a likelihood ratio. And then you have to multiply that likelihood ratio by the prior probability that this thing is true. So this requires some estimation. A prior is our estimate of some kind of bias. So in clinical tests, it's the estimate of the prevalence in the population. For example, if you give a COVID-19 test kit to someone, there is a rate of false positives and a rate of false negatives. And if you test enough people, you know you're going to get false positives and false negatives. And so the prior probability that you're testing someone who has the actual disease is dependent upon the prevalence of that disease in the population. And that will change how you think of the result you get when someone tests positive or negative. The thing that we know at the end of the test is not whether or not you have the disease, but the posterior probability after taking the test and either having tested positive or negative that you have or don't have the disease. In Bayesian reasoning, you can never really be certain of anything, but you can assign probabilities to almost anything. Let me give you an actual example. I think it's really helpful to think about this in maybe real-world terms. So when you do clinical trials, we tend to test things that we think might be effective and might be effective for a certain condition. So imagine that someone says they want to test Reiki against chemotherapy for treating cancer. Well, Reiki is one of those energy based therapies where they assume that there's some sort of cosmic energy that is flowing through all of us and that can be manipulated by the Reiki practitioner. Sometimes it's called like the God consciousness or cosmic energy. There's different words for it, and there's different kinds of energy treatments, many of which are similar to Reiki. Some people will pass like rods over your body or other things. In Reiki, they use their hands. So you might ask yourself, well, it makes sense to test it against something that we know is effective, at least to a certain degree, to see if it's just as effective. But if you think like a Bayesian, you may actually not want to test it. Because if you estimate the prior odds that an energy therapy, which is breaking the laws of physics, is actually going to be an effective treatment, then that might be a very, very small number. For example, you might say, well, it's about 1,000,000 to 1 or 1,000,000,000 to 1. And no matter what results you get, you still wouldn't believe them. So when it comes to actually doing like clinical trials, that even though this therapy is almost impossible, we're going to look to see if it works. There are actually tools you can use to estimate what's called the false positive risk. The false positive risk takes the alpha of your study and the beta. Those are like the type 2 and type 1 error rates, the chances of you rejecting the null hypothesis when you shouldn't, and the chance of you not rejecting the null hypothesis even though there is an actual effect. And then you modify those in this equation. I'm not going to say it on my podcast, but you can look it up, the false positive risk. You multiply those values by your prior, and that will give you the false positive risk. Now, if I had, for example, a chance of 1 in 1,000, let's be really generous to Reiki right now. Let's say there's 1 chance in 1,000 instead of 1 in a billion that it might work. Well, I can set my alpha at 0.05, which is what most studies do. I can set my beta at 0.01, and these are both reasonable things that studies have a lot. When I calculate it out using the equation, the false positive odds end up being 555 false positives for every 11 true positives. Or you could say there's a 50 times higher rate of false positives than there would be true positives. In terms of a probability, you have about a 98% chance of getting a false positive. So if I were giving something 1,000 to 1 odds, or 999 to 1 odds, that would mean that I would get way, 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 way more false positives than true positives. I may not even want to do the study because the chance that the positive I get is a false positive is super high. So that is Bayesian reasoning. But we know that people do these studies anyway. So what happens after the study? How do you calculate the posterior odds or the posterior probability that the result was real, even though you did the study and you probably shouldn't have? So the thing you can do here is you can look at the outcome of the study. For example, you find what your proposed magnitude of effect size was, and what are the confidence intervals around those. And from information like this, you can calculate something called the likelihood ratio. Now, a likelihood ratio is like an odds, and it's the odds or the likelihood that you got a true positive or a negative result. Now, just because you got a positive or negative result, that doesn't mean it's a true result. What you need to multiply the likelihood ratio by is the prior odds that that result is actually true. And that will give you the posterior odds. So basically, the equation is your posterior odds of something actually being true equals the likelihood times your prior odds. Let's just imagine that your prior is 1 to 999, like we did before. So that means that your odds would be 1 over 999, or 0.001001, etc. If you had a likelihood ratio that your results were true of 5, then your posterior odds would be 0.005005, which translates to a posterior probability of 0.49%. If the likelihood ratio was 1,000, extremely high, then your posterior odds would be 1.001001, and that translates to a posterior probability of 0.5, or about 50%. So you would have to have an extremely large likelihood ratio, 1,000, in order just to get a 50-50 chance that this result is actually true. Now, if I haven't lost you already, you're probably wondering, like, why are you telling me all this? What does this have to do with brain networks? Well, our brains are basically Bayesian calculators. They set prior probabilities that things are true. The brain has models, and it tries to fit your sensory data to that model, and it can adjust its prior probabilities. Some of these things are referred to as like expectations, or maybe the default settings or predictions that your brain makes. Now, network models especially can help us to predict how your brain behaves when different parts of it send messages to other parts. So network models are plausible predictions that have consilience with what we know about other sciences and the way that groups behave. So they provide really testable predictions of what our brain is going to do. In order to understand networks, it's usually good to start with something kind of very simple. And nothing in the brain is simple, but let's just imagine a very simple set of neurons. So most signals in the brain are not actually processed by a single neuron. They're processed by these networks of neurons. These networks in the cortex exist in columns, where you have like 6 layers in a column, and each of these layers have various kinds of neurons in them. Well, these neurons are glutamatergic or GABAergic. The glutamatergic neurons are pyramidal neurons, and they're the ones that kind of send and receive signals out of the network. And then inside the network, there are GABAergic neurons, and these GABAergic neurons are either inhibiting or not inhibiting these pyramidal neurons, or potentially even each other within this network. Pyramidal neurons can often excite each other. And so you end up getting a state where the network itself sort of ramps itself up. And we'll get to what that means here in a minute. But inside these networks, it's not just the signals that they're making there. They're also receiving signals. A lot of the signals that most of the cortex gets comes through or from the thalamus. And so you got the thalamus sending signals back to the cortex. But then you have your brainstem nuclei, things like the ventral tegmental area, which ships off dopamine to your cortex, and the neurons there will modulate the activity of the GABAergic and the glutamatergic neurons. You also have afferents coming from the raphe nuclei, which has serotonin and is dumping serotonin. I'm going to stop there with the brainstem nuclei. There's all sorts of other neurotransmitters that could come into play. But the point in what I'm saying right now is that it gets really complex. And it's so complex that we can't really understand what's going on. I mean, like, we can't handle the truth. I mean, even in a vastly oversimplified model, a human brain is really incapable of understanding the extent of the interactions that make human brains work. And so what we can do is model it. And one of the ways we can model it is using something called small world networks. Now, a small world network is kind of like what it sounds like. It's a small world after all. Don't sue me, Disney. But like, that just means that if you take a random person out of the entire globe and you pick another random person, and let's just say it ends up someone is in Saudi Arabia and someone else is in Ireland, you can connect those people in about 6 steps or less. So that just means that there are so many different connections between the people in the world that you only need a few steps to get from one person to another. Now, networks are made up of things called nodes, and nodes can be grouped into communities or cliques, sometimes they're called, and you can have hubs. So a node is something that sends and receives information, and the sending and receiving of that information is called an edge. You can have groups of nodes together and little communities of nodes. But if you have an area where there's tons of information coming in and out from a lot of different areas, that's called a hub. It is highly connected to other nodes and communities. So this gives the network something called scale-free properties. And it doesn't mean that the network can't scale up, but what it means is just that a small fraction of the nodes have a very large fraction of the connections. So those hubs have a lot of connections. The other nodes will only have a few. Most of them have a few. A few of them have a lot. There are major hubs in the adult brain. A few of them are like your putamen, the pallidum, so like your striatum, things going in and out of your striatum. Then there's the thalamus, the thalamic nuclei. It's kind of this joke, everything touches the thalamus. And then there's like the superior parietal lobe and the superior frontal lobe, parts of your central executive network that act as hubs with lots of connections to other parts of the brain. And there's a part of your brain called the precuneus, which is kind of in the medial part of your brain near the back. And it's a major hub of something called the default mode network. And then, of course, we have like our hippocampus, which is our memory hub. And so it has to be connected very widely in order to do its job. And then we have a hub in our insula that's part of something called our salience network. And then there's another hub for visual information that's called the left lingual gyrus. We're not going to talk about all of these hubs today, but I just want to mention them. That we have hubs and we also have nodes. And so you can model the brain kind of like a small world network. Now it gets this way because of genetics primarily. The broad network design, the way our brain is designed, starts with genetics. But the brain wires itself based on our experiences, like things in the environment as well, like the things that you eat. If you get infections, that might affect the way your brain wires itself. And then, of course, there's just some random chance involved in all of that too. So what that means is that neurons that fire together wire together. So as you are developing, if you get groups of neurons that tend to fire at the same time, they will also wire together and communicate together. I mean, think about how long it takes an infant to develop like the basic skills they need to just pick up a Cheerio and put it in their mouth. It takes a lot of effort. And then after some time, the neurons needed to actually do that, the local networks will wire together very strongly, and it becomes a lot easier. But it takes practice. So one of the things that make networks really cool is that they really make decisions by committee. So information comes into a network, and a lot of that information is lost when it leaves the network because it's integrated into maybe a single or just a few signals. So that means you can really model networks on different scales. So you have your neurons themselves and the networks of neurons, and you can take those networks and you can have networks of small networks. And then you can take those small networks and make bigger networks of those smaller networks. So this is where we get into like our whole brain function, things like our salience network and our central executive network and our default mode network. But then that whole brain integrates functions into what we do as a human. So you could say that our individual self is an integrated function of our brain. And so we can model ourselves and our behaviors and how those affect our environment, but also how those affect our own brains. And then you can have various people, and those people can form groups. Those groups or institutions have their own behaviors that can be modeled. So you get to this point where you have different levels of a network, and each level can be modeled pretty much the same way the smaller levels or the larger levels are, because they do the same thing. They take a bunch of information and then they integrate it into a few different things. Now, when you're integrating information into just a few different things, that means a lot of information gets lost. But it also means information can be gained. And usually that's misinformation. Like, so it's some random firing or some random signal coming from another place that actually doesn't mean anything. And so our brain has to deal with all of this stuff. It has to deal with things that have real information in them, things that are true, the true positives. It has to deal with information that is not true, the misinformation, the false positives. And then it has to deal with not getting information when there is actually information there. And that would be like a false negative. This gets us to what I told you we were going to talk about in the beginning, attractor networks. Attractor networks are simply networks, often small world networks, that create in themselves some sort of stable states where they exist. So there's usually two states at least in a attractor network. There's like a low energy state, and that's what keeps the network functioning. It's always at this low energy state unless it's doing something else. And it's sending out little signals, but those signals are just kind of random noise. And that random noise is being sent out because the system basically has to maintain itself. And it's not sending out a big enough signal that the other systems are going to actually read that as a true signal. But sometimes these networks get into a high energy state. So something comes into that network. It's usually a signal from somewhere else, but it could just be a false signal or a random problem. But usually it's a signal from somewhere else. And that puts it in a high energy state, and it tends to stay in that state. It becomes highly active and robust in that state. Then after a while in that state, it's going to go back down to its low energy state. If it doesn't, then that's a problem. So we call these things attractor basins. You have your low energy state, and then you need a certain amount of energy to get you into the high energy state. You could call that like the energy of activation. That's what they call that in chemistry when they're talking about what gets chemical reactions going. So you're in the low energy basin, some glutamate gets spilled into the synapse, and that is enough glutamate to put you into the high energy basin. So that's when you get excited. You become a high energy attractor network. And then in that network, you're maintaining your own activity and you're sending signals to other places. just saying all this stuff doesn't really make it real for anyone and is probably boring, and you might be thinking about just skipping this podcast right now. So let me relate this to something real that a psychiatrist might have to deal with. So I came across this paper which proposes that attractor networks might be able to explain some things like the positive symptoms of psychosis. And so the way that they explain it is like this. Prolonged sensory deprivation can reliably produce hallucinations in people. We know this. You just take away all their sensory information that they're getting and they'll just start to hallucinate things. But why is this? Well, the hypothesis is that this lack of information kind of lowers that activation energy so that smaller signals can get through. And this makes sense. So basically, this activation energy acts as a Bayesian prior. Is this signal real? It has to be a big enough signal that your brain's like, yeah, it's real. So the small signals don't get through. But this is a trade-off. So you will get false negatives where the signal should have got through, but your brain didn't let it through, so you didn't sense it. But if you're in an environment where there's no signals getting through, your brain might be like, well, I'm going to lower that threshold a bit. Lowering the threshold is going to let signals through, so you're going to get more positives. Some of those are going to be false positives. But you're also going to get fewer false negatives. You're going to let through signals that it would have missed before. Now this, if you're letting through signals and you're getting a lot more noise through, false signals through, might result in hearing things that aren't there, seeing things that aren't there. And this is all because your brain has basically adjusted its Bayesian prior probability, and this has allowed a lot of noise to get through. So let me summarize that real quick. There's a trade-off in your brain between getting false positives, which are the positive symptoms of psychosis, things like hallucinations or delusions, things that aren't real but that you sense, you think are real because your brain is telling you that they are, versus false negatives where you're not getting information that is available to you. The way that attractor networks can work is that they can lower the energy of activation to get to the higher energy basin where you become active and send a signal saying, hey, this is a thing. If you lower that energy though, you're going to let misinformation or just random noise through, and that might be hallucinations. So this in and of itself, makes sense metaphorically, but how might that actually happen in the brain? Well, I talked a little bit earlier about the GABAergic neurons and the glutamatergic. So the GABAergic neurons are things that inhibit things from sending signals generally, and the glutamatergic neurons are sending signals and exciting things, and they work in kind of a balance with each other. So maybe you are changing the balance of the GABAergic and glutamatergic signals. You might have less GABA signaling, which will reduce the energy required to enter your active state. But we know that in schizophrenia, they also have less NMDA signaling. How does this work together? By having less GABA signaling, the energy of activation goes down, which means you can easily get into the high energy state. But if you have less NMDA signaling or glutamate signaling at the same time, then staying in that active state might be more difficult as well. So what can happen is that you can shift from low to high energy states in a way that is very unstable. So the way that they said it in the paper was, a network with these characteristics will display loose and erratic jumps between low frequency resting states and high frequency active states. So basically, your brain is disorganized, sending a lot of false signals. Another thing that could affect this are levels of dopamine, because we know that dopamine affects like GABAergic neurons, and it can also have an effect on the glutamatergic neurons, but it especially modifies GABA signaling. And so if you have too much dopamine in certain areas, it might result in a lot of inhibitory signal getting through. which will stop signals from being sent. And if you have too little dopamine in certain areas, then you might let a lot more signals through than should get through. In Parkinson's, this is a problem because a lot of signals from your motor cortex are getting through, and this results in like rigidity and tremors because you have too little dopamine. But if your cortex is dysregulated, then your ability to think about things, to pay attention to things, to even form words for things can be really impaired because you don't have the balance between the glutamatergic and GABAergic inputs that you need to have healthy, stable attractor states. Another thing that attractor networks might affect are things like your affect or your emotions, your mood, and your levels of motivation. Our brain has mechanisms by which it motivates us, and it can calculate like reward values of things. So let's imagine there's a boy standing under an apple tree, and it's late fall, so everyone's kind of picked the low-hanging fruit off the tree, but they see way high up in the tree a beautiful red apple, and they're very hungry. How do they decide whether or not to take the risk to climb that tree to get that reward? So we think that this risk-reward system is very important in things like depressive disorders and in obsessive compulsive disorders, and can also just be offline or completely dysfunctional in some psychotic disorders. So we know that in depression, people are often amotivated. In fact, that kind of defines depression. There's like an absence of goal-oriented behaviors. There's a lack of energy. Anhedonia. People aren't seeking pleasurable experiences like they used to. I mean, even primary reinforcers like food are devalued, and people might lose weight and quit taking care of themselves. Mania, on the other hand, is kind of associated with excess energy and excess goal-oriented behaviors, and hedonism, which is where you look for pleasure, and they can assign excess value, excess reward to different things. Like if someone sees something online, like, oh, you should invest in this stock, someone who is manic is way more likely to like put all their money into this than someone who's depressed who's going to be like, I don't really care. Well, manic person is going to be making quick, uninformed plans for the future. So they think this has a lot to do with our limbic pathways and our cingulate cortex and our orbital frontal cortex, among other things, but I'm going to focus on that for now. So let's just say the boy sees the apple. So a signal is sent from the eyes to vision centers in your occipital lobes, but it's sent in two directions. One of those is called the inferior pathway, and the other is like a superior pathway. So it goes to the temporal lobe and it goes to the parietal lobe. In the parietal lobe, the things like the form of the object, the location, the size, the distance, those are all calculated. But in the temporal lobe, that object is given some sort of identity, and the features of the object, the salient features of that object, are identified. Well, in this inferior or ventral stream, you could call it, that information is then sent forward to your orbital frontal cortex and to your amygdala. So the amygdala is sort of like a emotional center, and your orbital frontal cortex is a center where you either determine that something has a reward or not, or a punishment value. So then the boy has a kind of emotional response to that apple. Saw the apple, identified the apple, saw how far it was, and is kind of thinking about what it would take to get to that apple. But also at the same time, kind of having an emotional response to seeing that apple. So the amygdala gets information from the inferior temporal visual cortex, and then it assigns some sort of affective value, you could call it, based on memories of what this object is and what has done for you in the past. And then it sends this information to your hypothalamus, and the hypothalamus is the part of your brain which can initiate the autonomic and hormonal responses. So it can get you kind of revved up, excited, ready to go. But at the same time, in your orbitofrontal cortex, your brain is assigning a reward value to this. So the orbital frontal cortex is saying, hey, this apple thing has a reward value. And the reward value can be based on many other things, right? So the history is, of course, important. Your brain has to know something has reward value, but some things are built in, and those are called like primary reinforcers. So like sweet things or like soft things like that. Your brain is pre-built to assign reward to those things. But the boy can't taste the apple. He just knows the apple is there. And he has in the past tasted the apple. So they've stored the reinforcement value of apple in the brain. And the orbitofrontal cortex is pulling that up. Now, if the boy was already full, he might look at that apple and it gets no reward value assigned to it. But if he's very hungry, it's going to get a reward value assigned to it. And so that's going to influence what he does. Also, the current emotional state, the state that the amygdala has helped to put you in, is going to determine whether or not that apple is rewarding to the boy or not. And this is important in things like depression and mania. There are also some top-down factors, like stuff that is happening in your central executive network, where it is kind of assigning a modifier to that value of the apple. For example, if someone had said to the boy right before he saw that apple, like, these are the best apples in the world, then his probably dorsolateral prefrontal cortex is going to be sending signals down to his orbitofrontal cortex and saying, yeah, very valuable. And so this is what advertisers try to do. They try to get this top-down effort in your brain to modify the value that your orbitofrontal cortex is giving to things. So once the orbitofrontal cortex determines the value of that thing, it sends this information forward to the anterior cingulate cortex. But it also sends it to monoaminergic nuclei via the nucleus accumbens, attachments to the ventral striatum and the thalamus. So indirectly, it also is activating the monoamines in your brainstem. So in that anterior cingulate cortex, it's receiving this reward value information. But now I'm going to stop right there for a second. We're in the anterior cingulate cortex, and it got its information from the orbitofrontal cortex. But I want to back up to having received that information in your occipital lobe and then sending it north, sending it up, sending it to your dorsal stream, the parietal lobe. And that's telling you where that object is and the distance that object is from you, things like that. That information is being sent to the posterior cingulate. So here we have information about reward value in the anterior cingulate, and in the posterior cingulate we have information about like location and size, everything that's kind of modifying the identity of that apple. Well, this information from the posterior cingulate gets sent forward to parts of the middle cingulate gyrus and processed there. So in the middle cingulate gyrus, it's able to say, hey, look, this thing is here. I'm going to start calculating what we would need to do to get that thing. And so the mid cingulate is sending out signals to our premotor cortex saying, hey, get ready. This is what we might need to do. At the same time, the anterior cingulate gyrus is sending signals to the middle cingulate gyrus and telling it how rewarding that thing is. Parts of the medial prefrontal cortex and the amygdala and the insula and the autonomic brainstem nuclei are getting information about the affective value of this thing. How does it make you feel? Is it something that makes you feel happy, sad, or neither? You know? And that is modifying the reward value. And in the middle cingulate, your brain is calculating what actions it needs to take to either gain the reward or at times to avoid punishment, but that's not what's happening right now. And it's calculating a risk-benefit ratio, like what is it going to take to get this, and is it worth the risk? So what it's doing is facilitating something called action outcome learning or memory. And while it's doing this, it's activating your premotor cortex, which is getting ready to move. Now, say that the boy decides to climb that tree. He gets up the tree, he grabs that apple, he comes back down. Or let's just say he gets a whole bunch of apples, like he gets a bucket of apples out of the top of the tree, and he comes back down and he starts to eat them. Well, at some point, the boy is going to satiate, which means that he's not going to be hungry anymore. And in the brain, the way this works is that the orbital frontal cortex quits sending reward signals to your anterior cingulate. And so it's not getting reward anymore. And the boy doesn't get excited when he sees the next apple in the bucket. But Dr. O'Leary, what in the world does this have to do with attractor networks or depression? So we know that people who are depressed tend to ruminate on things like defeat or hopelessness or guilt. They really lack motivation, they lack energy, and at times even their basic drives to care for themselves are impaired, and they just quit eating and they quit taking care of themselves. If you think back to Freud, he described depression phenomenologically as anger or aggression turned inward, instead of the aggressive impulse being outwardly focused. Now it's inwardly focused, and that's what he thought resulted in depression. Well, there was Carl Jung as well, one of Freud's students who went off on his own. He thought of depression as the depleted ego's experience of itself. So the ego has lost energy, and now it's experiencing itself in a very low energy state. Then there was Beck, and Beck is arguing that, well, we have these dysfunctional and distorted beliefs, and they can be incorporated into these self-defeating schemas. And when we activate these schemas because something happens in our life, we become depressed. And then there's Mahler. And Mahler is saying that depression happens because of an object lost. But the loss that you have isn't like losing a mother. It's a loss in fantasy. It's an imaginary loss, and this results in intra-psychic conflict. The difficulty with concepts like these is that they're metaphorical, they're untestable, and they really offer little help in elucidating like the neuropsychiatric mechanisms of the disease. However, network science is testable. There's this scientist named Edmund Rolls, and he works at the Oxford Center for Computational Neuroscience. And so he proposes in a paper that depression is a non-reward punishment attractor state that is initiated by our lateral orbital frontal cortex, and it spreads to our top-down, the executive networks, and then exerts a negative bias on our cognition. And this further biases our orbital frontal cortex and reward centers toward non-rewarding states. This means that the lateral orbital frontal cortex, which signals non-reward and punishment, gets increased in activity. The medial orbital frontal cortex reduces its activity, and it's the one that usually sends reward signals. And this biases our affective system as well, our mood. And it prevents us from actually participating in reward-seeking behaviors. Therefore, we become kind of helpless and hopeless. Now imagine for me two things. One is there's this bowl of soft serve chocolate ice cream that has a nice cone shape to it. Right next to it is a little turd that's shaped the same way. Well, if you were to see the turd on the table in front of you, most likely your lateral orbital frontal cortex would be the thing sending the signal. So this is like non-reward or even punishment. Like, this is a gross thing. I don't want anything to do with it. That's your lateral orbital frontal cortex. Your medial orbital frontal cortex will be activated more with the ice cream sitting in front of you. You're like, yes, sweet, yum, chocolate. But in depression, according to rules, the lateral orbital frontal cortex is biased toward false positive non-reward signals. That means for some reason, it's just like sending a bunch of false positive signals. Now, some people might be more predisposed to something like this than others. For example, like just because of genetics, we know that there's a large genetic component to mood disorders. It could be that chronic stress is contributing, or that there was a large adverse event. Also, things like brain lesions, for example, like tumors or even epileptiform activity can result in changes in mood. But it could also be lack of activity in the medial orbitofrontal cortex, which usually sends reward signals. And that is reinforcing the lateral orbitofrontal cortex's false positive signals, because the medial orbitofrontal cortex usually inhibits the lateral orbitofrontal cortex. Another thing that could be is that top-down control is impaired. So think about the advertising I talked about earlier. Your central executive network is supposed to be saying, yeah, this is great. But if it's not saying that, or if it's actually saying, why try, then that can bias the reward value of what you're sending toward the lateral orbitofrontal cortex. And what this looks like is rumination. You just think about things a lot and come to no real conclusions. You have guilt. You can even feel a sense of worthlessness. And this is all happening while the medial orbitofrontal cortex is biased toward false negative reward signals. So reward signals are not being sent even though they should be. And all of the same reasons that I talked about with the lateral orbitofrontal cortex might be contributing to why this is. So what this means, though, for the medial orbitofrontal cortex is more energy is needed to signal reward. But also less energy is needed to return to that low energy basin. So even if a reward signal gets through, you're very likely to just slip back down into the low energy state and stay there. So this results in things like anhedonia, low motivation, lack of energy, and eventually just hopelessness. Now, another way that attractor networks might affect people's levels of motivation is in obsessive compulsive disorder. And it also includes the orbitofrontal cortex. Imagine that the boy is eating apples, but he fails to satiate. He fails to get to the point where he feels like he doesn't need to eat any more apples. He's had enough. Well, in obsessive compulsive disorder, people often feel like no matter what they do, they haven't actually accomplished the goal, gotten the reward from it. And so they just do it over and over and over again. And they often feel like something really terrible might happen if they don't actually do this. So this could be because the middle cingulate cortex is continually repeating a signal to gain the reward and avoid some sort of punishment or loss. And this is constantly activating the premotor cortex. Remember, we're talking about the orbitofrontal cortex going to the anterior cingulate, which is then communicating with the middle cingulate, which is telling your body what to do, basically, how to get this thing. So this circuit just keeps going over and over and over again in some sort of high energy attractor state without any real reason to be there. So what's happening is there's this failure to complete the action outcome learning. That is the normal way your body works. This also can affect mood because the orbitofrontal cortex is not sending its normal reward signals to your nucleus accumbens when the object is gained. So if an object is gained, then you usually get this reward signal. Yay, got it. And that part of that is going through the nucleus accumbens. And then that's communicating through the pallidum to the thalamus, especially the kind of lateral habenula, which was sending this lack of reward signal, this false negative, you could call it, signal to your brainstem nuclei. And this is just reinforcing the whole failure aspect of what's happening. No matter what you try to do, it still feels like a failure. The overall result is really a failure of your action outcome learning. So you kind of perseverate on this thought, this urge, or this kind of imagined stimulus, and an action often that might help with that. But no matter what you do, you can't feel like it was complete. You're never satiated. Now, just knowing that we have these sorts of potential attractor networks in the brain and that these can help explain things like depression and OCD and aspects of psychosis, how does that help us? Well, right now, not a whole lot, but it can help us to understand why certain treatments might work. For example, in affective disorders, ECT is the most effective treatment we have. And it is associated with brain connectivity changes when you use it. So there are changes in the orbitofrontal cortex and the ventrolateral prefrontal cortex and ventral medial prefrontal cortex, which are right next to the orbitofrontal cortex, and also the dorsolateral prefrontal cortex. So when treatment is successful, these parts of these brain regions change in their connectivity to other parts. And the same is true when we give people TMS, or when we give people pharmacotherapies like SSRIs or SNRIs. Now, the review that I read didn't have a lot of information about psychotherapies, and the information it did have showed that they're really, even though there was predicted changes, they didn't demonstrate those changes very reliably using an fMRI scan. So we don't really know if psychotherapy is working through changing brain networking, but the way that it's conceptualized is that it's acting more on your central executive network. We also use drugs like ketamine, and ketamine blocks NMDA transmission, which means it blocks the excitatory transmission going through, which means that in effect, it is kind of increasing the amount of energy you would need to get a signal through. Now, in our depression model, this would mean that ketamine is making it more difficult for the lateral orbitofrontal cortex to send non-rewarding punishment signals. But we also know that ketamine's effects wear off. They are temporary. So a few days after you give ketamine, your attractor states just return to what they were before. So you have to repeat the treatment. And of course, we have our serotonin reuptake inhibitors, our serotonin and norepinephrine reuptake inhibitors. These are the first line of pharmacotherapy for depression. So one of the proposed mechanisms is that during a depressive state, you have these like false positive, non-rewarding or punishment signals being sent by the lateral orbitofrontal cortex. And this is resulting in the lateral habenula sending a signal down to what's called the rostral medial tegmental area. And that area has GABAergic neurons that go to the ventral tegmental area and the raphe nuclei. And those GABAergic neurons reduce the production or the release of dopamine and serotonin. So by interrupting these signals from the orbitofrontal cortex, by reducing the excitability of the pyramidal neurons there and increasing the GABA transmission in the cortex, you are normalizing the brain's ability to produce monoamines. You are countering this suppression of monoamines from the lateral habenula, as well as probably quite a few other things at the same time. Now I want to take a little time to kind of wrap things up a little bit. So remember earlier I was talking about how there are these different levels of networking. We have like levels at the neuronal level, these groups of neurons together, and then those can form larger brain networks, and those larger brain networks come together to do whole brain activity. And then that's what makes the basis of the individual and the individual's behaviors and their actions. But the individual is also parts of groups in society, and those groups and institutions themselves have actions. And all of this feeds back on each other. So for example, if there are institutions causing people social distress, then that's going to result in individuals having affectual and behavioral reactions to that. And that's going to affect the autonomic nervous system and our monoaminergic system, which itself is going to affect those networks and change the attractor states of these networks in our brain. So no matter where you enter this system, you're going to have an effect on probably every single other layer. So psychotherapies are supposed to hit at the like behavioral and thought part of an individual. And if you can kind of change your behaviors and change your thoughts, this is thought to affect things like your autonomic nervous system and even the release of monoamines, which can change your attractor states in your brain. So it's kind of a top-down way to get there. There's even a much higher top-down state where we have institutional or cultural changes. For example, like for psychosis, we might institute things like assertive community care or having availability of care being there for people to get. And this can affect the individual and on down. So what I want to accomplish today is to introduce you to the idea of networks in the brain and how these networks can be modeled as attractor networks, which means that there's information coming into them and going out. The information coming in is oftentimes just noise, and so you don't want to move from like a low energy attractor state to a high energy attractor state unless you're very confident that it's a real thing. And the way that the brain sets that confidence level is through the energy of activation that it takes to get from the low to the high energy state. It's like a Bayesian prior probability built right into your brain. Once you get to the high energy attractor state, you also need to be able to come back down. And it takes a little bit of energy to do that. Usually that energy is just provided by like the random noise that you have there and the GABAergic inputs. So it just keeps the attractor state from revving itself up and staying revved up forever. When any of these things are out of whack, you can either stay in a high attractor state for way too long, or you can have like instability of just going back and forth between different attractor states. Your brain can also make its own adjustments. It can try to reduce the energy it takes to get more information moving through the brain, but this is going to result in more false positives, even though it's reducing the number of false negatives. I really think that as psychiatrists, we need to start thinking more like a Bayesian. We need to consider things like prior probabilities and likelihood ratios instead of just looking at things like, well, it has a p-value and it passed the test. Well, that does not tell me whether or not the results are true. That just tells me whether or not it passed a statistical test. The only way to know whether or not the results are true or high confidence instead of being true, because you'll never really know. You'll never know if something is true or not, but you can have high confidence in it or low confidence in it. So instead of just looking at p-values, we need to be exploring things like the likelihood ratios and what that means if you set the prior probability at different levels. If you can set the prior probability low and the posterior probability is still pretty high, that gives you a high level of confidence that the results were real, instead of just arguing over the size of our p-values. Thank you for listening. I am Dr. O'Leary, and this has been an episode of Psydactic Residency Edition.